Good morning, my dear friends. Welcome back to my YouTube channel, Medical Classes by Dr. Srinidhi Kumar Acharya. Please don't forget to subscribe to my channel and also please give your valuable comments. You can also visit the playlist. In the playlist, I have provided almost all the topics in a very systematic order, and this is quite convenient for the study. I hope you will make use of it, my dear friends. We have been discussing about the bleeding and clotting disorders in children. We already covered three parts, and I have tried to give you some basic information regarding bleeding time, clotting time, factor associated, etc. Now, today onwards, we shall start in detail explanation of the bleeding and clotting disorders. First, we will understand the bleeding process of coagulation and the role of the coagulation factors, and in the coagulation cascade, what do you mean by intrinsic pathway and extrinsic pathway? What is the basic of this? What is the basis of coagulation? How it works and how it discovered? We all see all these details in today's class. To begin with, we shall have the introduction to the clotting factors. As we know, we have got totally 13 clotting factors. And factor number 1 is called as fibrinogen, and deficiency of the fibrinogen will going to cause a disease called as afibrogenemia or dysfibrinogenemia. Totally there is absence of factor number 1. Okay, this is possible in these two conditions, rather dysfibrinogenemia or afibrinogenemia. Second important clotting factor 2 is prothrombin. Okay, so prothrombin deficiency will lead to many problems. We know that especially extrinsic pathways get affected. And then comes the fourth one, that is proconvertin. Proconvertin is also called as calcium. Then comes fifth one, that is the labile factor. It's also called as proaccelerin. Absence of this factor or deficiency of this labile factor or proaccelerin will going to cause a condition called as parahemophilia, that we will discuss later. Seventh one is a stable factor, or it is also called as proconvertin, and that also leads to coagulation factor deficiencies. Now, eighth factor number 8 is called as antihemophilic factor. It's also antihemophilic factor, and this absence of this factor number 8 is also called as classical hemophilia or hemophilia A. Whereas ninth factor is called as Christmas factor, and that's called as very important factor, and absence of this factor or deficiency of this factor will going to lead to hemophilia B. Then 10th one is stored power factor, that leads to so many coagulation deficiencies. 11th factor is called as plasma thromboplastic antecedent factor. Absence of or deficiency of this factor leads to hemophilia C. 12th factor is called as Hagman factor, and 13th factor is called as fibrin stabilizing factor. Actually, there is one more factor which is also comes under the concept of platelet adhesion, that is the von Willebrand factor. Von Willebrand factor is factor 8 stabilizing factor. Factor 8 is anti-hemophilic factor, and the hemophilic factor is factor number 8. But to stabilize this anti-hemophilic factor, we need another factor, and that is called as von Willebrand's factor. So these details we will see in coming classes. Okay, so now we shall have an introduction on interpretation of coagulation profile. What is this coagulation, how the clotting takes place, and what is their importance? So this understanding of these basics is very important. Now, as we know, there are mainly two pathways related to coagulation. One is intrinsic pathway and another one is extrinsic pathway. Okay. Normally, you just imagine, normally inside the vessel, blood will not go into coagulate, isn't it? The blood is flowing inside in a normal person, physiological condition I am talking. In a normal person, blood is present inside the vessel, so there is no question of any coagulation at all. But as soon as we bring this blood outside into a test tube or in a syringe or anything, now the clotting starts. Okay, So to prevent this coagulation, when you take the blood outside, you have to add some of the anticoagulants. If suppose you are sending the blood to the investigations, etc. So inside the vessel, blood is not clotting, but as soon as the blood comes outside, it starts clotting. That means when the blood comes out, some non-endothelial surface. That means, see, in the blood vessel we have got an endothelial surface. I can interpret it in a different way. As far as blood is in contact with the endothelial surface of the artery, the blood will not go into coagulate. But if the blood is removed from the endothelial surface, that means no more endothelial surface is available for the blood, then the process of coagulation starts. So that means when the blood comes out of the some out and contact with some non-endothelial surface, the factors present in the blood get activated and the coagulation begins. Something happens when blood is removed from the endothelial surface. As soon as the blood touches any non-endothelial surface, what happens is some factors are get activated, and as a result, the blood will undergo clotting. Coagulation begins. So initially, when scientists were trying, during the discovery of these factors, these factors which is responsible for this bleeding coagulation, when the blood is taken out of the endothelial surface, so they are basically called as contacts. Okay, contacts. Just like factor, they call it as contacts. And some doctor gives their personal name also for that, like Hegman's factor. Okay, as they're called as a and they call this as intrinsic pathway. Okay, so blood when comes outside the endothelial surface, it starts clotting, and they call this as intrinsic pathway. ये जो प्रोसेस है ब्लड का जो क्लॉटिंग है, ब्लड को आप बाहर लेके आ रहे हो, जैसा ब्लड को वेसल से बाहर लेके आ रहे हो तो उसमें क्लॉटिंग शुरू हो जाएगा. ये क्लॉटिंग जो शुरू होने का ये जो प्रोसेस है, इनिशियली साइंटिस्ट ने क्या बोला कि दिस इज कॉल्ड एज इंट्रिंसिक पाथवे. इंट्रिंसिक पाथवे. नाउ एक बार ब्लड को बाहर लेके आ गए उन्होंने, अगेन दे कीप द ब्लड आउटसाइड एंड दे पुट सम citrates, for example sodium citrate, inside it and they mix it. Now we know that sodium citrate is negatively charged. Okay, so what they observed is when they put the sodium citrate into this blood which is already taken outside, the process of coagulation is going to stop. पहले जो coagulation हो रहा था वो बंद हो गया. Now they again have got another interpretation. What is that? When the blood is taken out, it starts coagulation as it comes out of the endothelial surface. But when you add any substance which is negatively charged, especially like sodium citrate, the process of coagulation stops. That means the sodium citrate will go into block or neutralize some of the factors which is actually causing the coagulation when the blood is taken outside. Okay. Afterwards, they again added some tissue juices. Okay. Tissue juices, for example, you take thromboplastin. Afterwards, again they added some tissue juices. For example, thromboplastin. The coagulation again starts. पहले ब्लड को बाहर निकाला था, coagulation हो रहा था, उसको रोकने के लिए सोडियम सिट्रेट को ऐड किया जो नेगेटिवली चार्ज था. शायद उसने जो है वो जो क्लॉटिंग कर रहा था जो फैक्टर्स उसको ब्लॉक किया या न्यूट्रलाइज किया, क्लॉटिंग बंद हो गया. क्लॉटिंग बंद हो गया. दिस इज व्हाट वी फॉलो व्हेन वी आर स्टडीइंग द इन्वेस्टिगेशन टू द लैब. ओके, बट उसके बाद अगेन दे एडेड सम टिश्यू जुसेस. फॉर एग्जांपल यू टेक थ्रोम्बोप्लास्टिन, अगेन द प्रोसेस ऑफ coagulation started. Okay, now they call this pathway as extrinsic pathway. They call this pathway as extrinsic pathway. Detail we will see again. This is just a superficial knowledge. Okay. Now, when the injury is taking place, what happens? Suppose injury is taking place, bleeding is occurring. What actually happens? Actually, both intrinsic and extrinsic pathways are get activated. Whenever there is an injury to the blood vessel and the bleeding is going on, in that time, both intrinsic pathways also get activated. Extrinsic pathway is also get activated. Now you know that intrinsic pathway means blood when the blood is come out of the endothelial surface, some of the factors which are present inside the blood, they now get activated, alert, and start the process of coagulation. So that is called as intrinsic pathway. What do you mean by extrinsic pathway? When the bleeding takes place, when you add some tissue juices inside it, okay, tissue juices like factors like thromboplastin inside it, again the blood starts coagulation. So this is called as extrinsic pathway. So what I mean to say is, when injury is taking place, both these pathways are get activated. Okay, when there is an injury, the blood meets the endothelial surface, subendothelial layer, and it activates the platelets. The factor number 12 will be activated. This leads to the triggering of the intrinsic pathway. So in general I told. Now what actually happens, how this intrinsic pathway is activated, I told that as soon as it comes out of the endothelial surface, this happens. But as soon as blood comes out of the endothelial layer or subendothelial layer, the first thing which takes place is activation of the platelets. We have already studied. And as soon as the activation of the platelets takes place, the factor number 12, clotting factor number 12, okay, so that is the first which will going to activate. Once the factor number 12 is get activated, clotting factor number 12 is get activated, it leads to the initiation of a intrinsic pathway. Factor number 12, you know here, factor number 12, Hagman's factor. Okay, so when this is factor number 12 is first initiated, triggered, when this is activated, then it leads to the formation of cascade of changes in the intrinsic pathway. Now, similarly, there is also the activation of the extrinsic pathway. Okay, now extrinsic pathway means when you add some tissue factors or tissue juices, the coagulation begins. If you add sodium citrate, coagulation stops. Okay, now similarly there is also activation of the extrinsic pathway. Tissue factors is with just tissue factors or tissue juices. They are actually nothing but a protein, or it may be a lipoprotein. Okay, so tissue factors, probably I told thromboplastin. So thromboplastin, so tissue factors like thrombofactins are present in the blood. These thromboplastin like factors, first they will go into activate the factor number 7. Factor number 7. There in the intrinsic pathway, first activation is factor number 12. Here the first activation is factor number 7. So once the factor number 7 is activated, it triggers the extrinsic pathway. That means factor number 7 is activated. Factor number 7 is activating probably some thromboplastin like substance, because thromboplastin is very important for the clotting in the extrinsic pathway. So as a result, thromboplastin is formed and the extrinsic pathway begins. Okay, but it may be intrinsic pathway, but it may be extrinsic pathway, but the end result is same. End result is what? Both is causing the coagulation. Intrinsic pathway is also intended to cause the coagulation. Extrinsic pathway is also intended to cause the coagulation. The final aim of the both pathways are same, but extrinsic pathway is a little faster pathway. Okay, extrinsic pathway is a little faster pathway. Directly factor number 7 is activated, and then prothrombin, and then comes the fibrin, fibrinogen, etc. Whereas intrinsic pathway is quite longer and slower pathway. Both will go into cause coagulation. Okay, so but it could be an intrinsic pathway or extrinsic pathway. The end will be always same. That is conversion of the soluble fibrinogen into insoluble fibrin to form the coagulation plug. Okay, I hope you understand. Now, meantime, the liver also produces a lot of coagulation factors round the clock, and it releasing into the blood. A lot of clotting factors are produced from the liver. Again, the details we will see in coming classes. The factor number 12 is the first to react with the endothelial membrane when it touches and convert it into an active form of factor number 12. And this factor number 12 now activates the factor number 11. And factor number 11 now activates the factor number 9. So this is how the cascade of changes take place. Okay, I told how the intrinsic pathway begins. Intrinsic pathway begins with the initiation or stimulation of the factor number 12. Okay, so liver is producing the factor number 12, around the clock it is producing, okay, it is flowing in the blood also, factor number 12, but when this factor number 12 reacts with the endothelial surface, okay, it touches the endothelial surface, it converts into its active form, and active form of factor number 12, and that will go into activate the factor number 11, that will go into activate the factor number 9. So this is how again the intrinsic pathway begins. Comparatively this is a slower pathway. Okay, now again to continue, once the factor number 9 activated in the presence of phospholipids, calcium and factor number 8 are get activated. Factor number 8 can activate the factor number 10. Okay, it started from 12, 11, then comes 9. Now again the factor number 8 is activated, and then it activates the factor number 10. Now once the factor number 10 is get activated, it converts the prothrombin into thrombin. Okay, prothrombin into thrombin. Now thrombin will break down the fibrinogen into fibrin monomers. These fibrin monomers will now go into deposit on the activated platelets. Activated platelets are also there causing adhesion and aggregation in the bleeding site. Now over that, these fibrin monomers go and get deposited and form a mesh-like structure, forming a strong hemostatic plug. Okay. Now one thing is very important, that is the presence of the calcium. For the correct formation of proper running of the intrinsic pathway, calcium ion is very, very important. As far as calcium ion is present in the blood, so that will going to cause the coagulation. If there is less calcium ion, then the coagulation is also get affected, because this factor number 8 activation, it requires the calcium. So it plays a very major role. Okay, calcium metabolism is maintained by parathyroid gland and thyroid gland. So in case of thyroid and parathyroid gland problems, again the coagulation problems will be there. The reason is parathyroid hormone is very much important for the regulation of the calcium. So this cascade. Calcium is very important for the activation of the clotting factors. Okay, so parathyroid gland problems and thyroid gland problems often leads to clotting factor deficiency and problems. Now, meanwhile, we know that thrombin is formed, isn't it? Once the factor number 10, we are talking about intrinsic pathway. Once the factor number 10 is get activated, then it converts the prothrombin into thrombin, and thrombin will break down the fibrinogen into fibrin monomers. So once the thrombin will be formed, once the thrombin is formed, now it also activates the thrombin stabilizing factor. एक बार thrombin बन जाएगा, तो thrombin also activate a factor called as thrombin stabilizing factors, which is also called as factor number 13. Factor number 13. Factor number 13 is called as fibrin stabilizing factor. Okay, it is also called as fibrin stabilizing factor. So now this is a fibrin stabilizing. So this will now rush to the fibrin monomers and cross link the fibrin monomers. इसका नाम ही fibrin stabilizing factor है. Fibrin बन गया, mesh जैसे बन गया, उस mesh को और cross link करके उसको और थोड़ा strong बनाना ही factor number 13 का काम है. So thrombin बनने के तुरंत बाद there will be also formation of factor number 13. Now this will be a strong meshwork coagulant-like structure over the platelets plug. Now this is called as primary platelet plug, which is further converted into secondary platelet plugs. Now at the end, when this factor number 13 is also get activated by cross-link the fibrin monomers, then it is called as permanent hemostatic platelet plug. Okay, so this is all about the intrinsic pathway, how it works. Now talking about the extrinsic pathway, in the meanwhile, there will be production of the tissue factors from the injured area. Injured हुआ है, bleeding site in the vessel. So some of the tissue factors are produced. We are not discussing about the extrinsic pathway. Okay, tissue factor from the injured area. So when there is production of this tissue factor, probably thromboplastin, so these tissue factors will convert the factor number 7 into active form of the factor number 7. What is factor number 7? I hope you know that factor number 7. Yes, stabilizing factor, pro-converting. It is called as pro-converting. It's called as pro-converting. So there will be production of the tissue factor from the injury, that's pro-convertin. This tissue factors will convert the factor number 7, that is pro-convertin, into the active form of the factor number 7. Now this active form of the factor number 7 activates the factor number 9 and factor number 10. So both will be now activate the prothrombin factors, which produce the thrombin and form the fibrin monomers and forms a hemostatic platelet plug. So this is how the extrinsic pathway work. Now extrinsic pathway is a shorter one and faster one. वहाँ पे क्या होता है, सीधा 7 फैक्टर नंबर 7 एक्टिवेट हो गया, उसने फैक्टर नंबर 9 को स्टेबलाइज किया, एक्टिवेट किया, फैक्टर नंबर 10 को एक्टिवेट किया, प्रोथ्रोम्बिन बन गया, फाइब्रिनोजन बन गया, फाइब्रिनोजन को फाइब्रिन बना दिया, मेश बना दिया, हो गया. It's very short. So intrinsic pathway is a little slow system, while extrinsic pathway is a fast system of activation of the platelet plugs and hemostatic plug, or formation of the strong platelet plugs or hemostatic plug. Now this is strongly sealing. ये जो mesh बन गया, now this is going to strongly seal the bleeding points in the endothelium, and the blood leakage will going to stop. In normal condition, the endothelium is anticoagulant, isn't it? Normally blood जो है clot नहीं होता है vessel के अंदर, because endothelium उसका है. जब तक वो endothelium contact में है, कुछ नहीं होगा. इसका मतलब है endothelium is normally anticoagulant. And is also anti-platelet adhesion. Normally platelet का भी adhesion नहीं होता है. But the same when it is injured, it becomes a procoagulant, and also it is a coagulant platelet aggregation. Now, meanwhile, when endothelium is injured, it also produces a factor called as, this is also very important. When endothelium is produced, we have already discussed this, it also produces a factor which is called as von Willebrand's factor, which will going to stick the plug in the endothelium breakpoint and increase the platelet adhesion. So von Willebrand factor is also required for the proper functioning of the platelets and forming the aggregation, etc., platelet aggregation and activation. And it is also required for the factor number 8. Factor number 8 का stabilize करने के लिए भी von Willebrand factor is required. So it is both in platelet disorders and clotting factor disorders. Von Willebrand factor is a common factor. It's required. Okay. So platelets which are now adhered will bring the granules like alpha and delta. What happens next? Now platelets which are adhered there, so they will bring the granules like alpha and delta, which brings the more platelet into the site and cause the vasoconstriction and cause the platelet aggregation. Totally the hemostatic plug will be converted into a strong plug within 1 or 2 minutes. So coagulation screen, let us discuss about the coagulation screen. So this is used as initial screening test in children who have the suspected bleeding or clotting disorders, or any cases before surgery, those who are on anticoagulant, and in certain disorders like liver disorders. So about the coagulation screen, we will go to discuss in the next class. Thank you very much.